technical issue. So hello, everybody. I hope you can hear me and see the screen. So it's my real pleasure to start this meeting. I just counted, we actually organized it. It's time number 8 since 2013 when we first, made it first time. So some of you many of you may know, but some not what MURI means in the title of the talk. It stands for Multi-Disciplinary University Research Initiative. And this is a program by Department of Defense, which supports basic science research. So we are very fortunate to obtain a grant from ONR, Office of Naval Research, in 2013 to study sleep and memory consolidation through that program. And that was kind of starting point for those meetings, which I think turned out to be quite successful over the years. Of course, this year is kind of very unusual circumstances, and I should say it was a little bit late decision to go forward with this meeting. Normally, would never be able to organize it, I guess, on such a short notice, but with this virtual format, it seems to be working quite well. I'd like to use the opportunity to thank Tom McKenna, our program director, who started all this kind of line of activities, including great research, I think, which involved many labs over the years. Actually, it's kind of expanded beyond the original scope of lab and the original scope of the project. So just kind of moving forward. So if you see my screen, this is a program, and we have a little bit different format from normal presentations in the sense that we'll have 30 minutes talks, and we don't have explicit time for questions right after each talk. Instead, please go ahead and submit your questions in chat. And you can see we have two sort of moderated sessions, discussion sessions after each block of 4 talks. And during those discussion sessions, which Terry kindly agreed to run as a moderator, we essentially will have all the speakers, including those who haven't spoke yet during first discussion session, sort of be available to answer questions, to have a discussion, and so on and so on. So again, historically, this meeting was focused on sleep and memory consolidation, and this is kind of a topic of the first session, but we decided to expand it this year to include another exciting topic related to question how brain does computations using spikes and sort of techniques and approaches which are different from what I guess lots of artificial systems are using. So our session 1 is about sleep and memory consolidation, and I'm not planning to do any big introduction about that, but just in one sentence, essentially, sleep is kind of a mystery state of the brain, and what it does, probably lots of important things. And one thing for sure, we learned that it doing a lot about kind of organizing our memories and making them in a format which is good for storage, good for retrieval, and so on and so on. So this is kind of general topic, and this is direction of active research in many labs, and we have a number of very exciting talks today on this topic. So saying that, let me introduce our first speaker. We are very pleased to have Lisa Marshall from University of Lübeck. She speaks from Germany. We tried to bring her several times to this meeting, but I guess overseas travel before Christmas was always a little bit an issue. But this year, again, for good or for bad, this format kind of allows to do lots of things which wouldn't be impossible to do normally. So it's my pleasure to introduce Lisa, and she did lots of exciting research about memory and learning and role of stimulation in sort of promoting memory consolidation. So with that, I will stop sharing, and Lisa, if you can start sharing your screen, and please go ahead with your talk. You're muted still.
Thank you, Maxim. So do you see my screen?
Yes, we do.
Okay, good. Yeah, so I was going to give more or less a chronology of what we've been doing in relation to sleep and memory research, and building up also to how we got where we are. And one of the first things we started with, which is also an introduction basically into just the general sleep cycles that one has, is that instead of only measuring the polysomnography, which gives you this nocturnal, let me get my pointer up here, sorry. Instead of just showing you for humans here, we have this histogram of sleep where we have the non-rapid eye movement and REM, rapid eye movement, sleep, and the different 4 to 5 cycles that one has in the course of the 8 hours. This is basically derived from the EEG. And in addition to EEG, your brain also produces very slow potential shifts. And that is what we started out measuring a while back already, and saw that this non-REM REM sleep cycle is associated with quite unique shift in the direct current potentials, which yeah, basically was characteristic for the cycle. It was also, or not only, associated with electrophysiological events, but reflects basically the activity going on in the brain at other levels, for instance, hormone secretion, so that if we here look at, this is the cycle that we see over here, here we see that in the middle of the cycle, there is an increased peak of this potential, increased positive peak, and that's the time when the luteinizing hormone secretion was initiated. And so we asked ourselves what these potential shifts indeed reflect. And one way of possibly doing that, that's in itself a hypothesis actually, is to see what happens if you apply constant current to the scalp. We're not one of the first ones doing that. Others have done that long time back in history. Also in the 80s, there was a group who investigated that, also the biophysical associations quite strongly. And so based on their parameters, we then applied during this deepening of non-rapid eye movement sleep, transcranial direct current stimulation, so nodal stimulation, positive polarity during this time point of sleep. Before this, we had the subjects learn and afterward recall memory tasks. So basically, one of the functions of sleep is postulated to be memory consolidation. And so we wanted to see if this interaction with a DC potential, if that would affect the memory consolidation. And I won't go too much into this study, but just to show quickly the results, that we did see there was an increment after the stimulation, after the TDCS, in the recalled word pairs. So measuring the difference between recall and learning, we saw an enhancement in the number of word pairs that was recalled. That is a declarative task, which other studies had at that time, pretty much in the beginning of that time, it showed to be associated more with slow wave sleep, which is when we applied the stimulation, whereas a non-declarative task did not benefit from this stimulation. On the right, we see then more or less that there was also a bit of a deepening of slow wave sleep, the time spent in slow wave sleep through the stimulation, the thick line, and some changes then also during acute stimulation in the power of slow oscillatory activity. And that, together with some other input, gave us the idea of not just applying constant stimulation, but oscillatory stimulation, which endogenously exists during the time of non-rapid eye movement sleep. And by doing this, and go through that also quite rapidly because it's been published, in this procedure, then we also saw that during the time period when stimulation was given, that is actually in the intervals between the stimulation, between the 5-minute blocks of stimulation, to be precise, that we saw that the slow oscillatory activity, the slow oscillations, their power was enhanced, as well as power in the sleep spindle frequency range. And that again, there was an increase in memory retention of the declarative task, here on the one hand for words, and then also for figures. We applied the stimulation in different paradigms, so to speak, where we measured on the one hand, this is what we just talked about, the effect of slow oscillation stimulation during slow wave sleep, then in a more or less paradigm where we wanted to see if it really was this slow oscillatory character of the stimulation that would or did enhance memory consolidation. We applied a frequency, theta frequency, which does not normally occur during deep non-rapid eye movement sleep during the same period of non-rapid eye movement sleep. When the subjects go into non-rapid eye movement sleep, into slow wave sleep. And here indeed we saw that there was a decremental effect on the retention of word pairs. And another paradigm we stimulated then during rapid eye movement sleep with theta stimulation. Here there was no effect on consolidation per se. There was, however, interestingly, an enhancement to some extent. One can argue about it, but there was enhancement in fast, more like gamma related rhythms that occurred. And as you probably know, theta and gamma oscillations are often linked in their occurrence. And then finally, we applied also this low oscillation stimulation during wakefulness, first as a control, and then later based on the preliminary results that we had is in a real experiment, and saw here, and that was based on the results that we had in this preliminary experiment, that this slow oscillation stimulation applied during wakefulness had not an effect on the sleep spindles, but had an effect on theta activity. So basically increased the theta activity. And as you probably know, theta activity is then ongoing in an enhanced way during learning, during higher cognitive activity. And so in parallel to this stimulation during wakefulness, we had a learning task, and here saw that there was indeed also an increase in learning performance in this design, this study. So just as this background again, or more getting more deeply into the memory consolidation, the general concept that exists, one main concept that exists on how or why memory consolidation occurs during sleep is essentially that oscillations which reflect the activity ongoing in different brain structures, that they reflect the interaction of these different brain structures, specifically the hippocampus, the sharp wave ripples in the hippocampus, sleep spindles in the thalamus, and the slow oscillations basically generate in the neocortex. That is one main concept. Another concept for the relevance of slow oscillations per se in the cortex is that they are involved in synaptic downscaling. So basically a depotentiation of synapses occurs in the course of the night. And in collaboration with another group, we then investigated with our data that we had already obtained to what extent there would also be a increased deceleration of the slow wave, the endogenous slow wave power after stimulation had occurred as compared to a sham situation. And indeed, it was the case that with this enhanced, or with the slow oscillation stimulation, that there was an enhanced decay, indicating more or less that this depotentiation had also been facilitated, or the reflection of it. So going to another study that occurred in parallel in the group that I was in at Björn Born, was a study where one investigated to what extent this downscaling, the synaptic downscaling, that would be then beneficial for learning after sleep. And that was conducted with an afternoon nap. And as the study showed, that the learning after stimulation period did then lead in some of the declarative memory or declarative encoding tasks then to an increment in learning performance. And that this increase in learning performance also coincided with an increase in slow wave activity, or coincided, at least there was during the nap, also an increase in slow wave activity that occurred with this slow oscillation stimulation. Now, the stimulation was not always as effective as we had initially thought. Here, basically, or our studies where we had wanted to compare actually the effects of nodal and cathodal stimulation, and first wanted to investigate how the coupling was influenced by this nodal slow oscillatory stimulation. And here we see basically the coupling, so looking at the mean vector length that we had for, let's just focus on the fast spindle activity. So how the fast spindle is coupled to the slow oscillation, here to the endogenous slow oscillation, which has a very high mean vector length. And then the effects that we had with a nodal stimulation, it was significant as compared to a shuffled sham condition. But as we see here, the strength per se was actually not that strong. And if we look at the effects of cathodal stimulation, then we see that here there was actually no significant effect. And interestingly, also that the phase coupling of the spindles to the slow oscillation was indeed at a different phase. Okay, it's not significant, but it was, it did not more or less coincide with the endogenous rhythm. And as you probably know, that in the course of the time, there were some studies that did replicate the experiments that we had, some studies that didn't replicate the initial study that we had. And I'll come to that later. As an interim, so that one can work a little bit more with the stimulation per se, we had a collaboration with Hartwig Siebner, who is now in Denmark, who is an expert in, or is an expert in TMS, transcranial magnetic stimulation studies. And basically we operated with the same stimulation paradigm, but here we could gather results more quickly in order to see the effect of the different stimulation patterns, which parameters might be effective and not effective. And basically these are the results that we saw there, that there was indeed a difference of the effect of a nodal and cathodal stimulation, however, that there was also a time-dependent effect that over increased number of stimulations, there was an increment in the difference of the effect of cathodal anodal stimulation on the amplitude of the motor evoked potential. And furthermore, that there are certain baseline influences. So basically, we saw that at an individual level, the amount of online facilitation of the motor evoked potential at the beginning of the stimulation, that predicted then the later offline effects. So there was definitely a carryover effect over a certain time period, which was the case for slow oscillatory stimulation as well as for the constant TDCS, which is more typically used for Loren stimulation procedures. So I think I'll skip over this one and come to how more looking into which baseline effects may influence the efficacy of stimulation. And in order to investigate this, we conducted an experiment where we have, on the one hand, a sham condition, which only involves a learning of declarative and non-declarative tasks. Here, the subjects slept in the morning, they recalled, and we only had more or less a few intervals of stimulation. Then we had a stimulation session where we applied slow oscillatory stimulation during non-rapid eye movement sleep. And then in addition, we had a control task where there was non-learning. The subjects watched a film. And so here the idea or the concept was that would learning, the change in the EEG that is associated with learning, so there was always a change in post-sleep EEG associated with learning to some extent. If this change from the control to the sham control to the sham session, changes in the EEG, if they would in some way relate to the efficiency of slow oscillatory stimulation on learning performance or memory performance. We investigated through time periods, the acute time period during stimulation and the 150-minute post-stimulation period. And here we found that if you compare the sham stimulation with the control stimulation, that there was a modest increase in the slow spindle density, particularly during the longer time period, the 150-minute post-stimulation time period. And unfortunately, when we compared all subjects, the effects on learning between sham and stimulation, there were no effects on stimulation across all of the subjects. Since we had wanted to investigate the effect of or differentiate between individual performance, we had also used a memory task, or rather a task to assess the memory quotient, which is a more trait-like associated feature. And here we saw that if we made a median split between the subjects, so differentiated them between the high memory quotient and the low memory quotient subjects, then indeed there was, for the high memory quotient subjects, an effect of slow oscillatory stimulation on their performance, that indeed there was a significant correlation between the efficacy of the stimulation on memory consolidation and the memory quotient. So, and how is this effect associated then with the EEG, or was it associated with any differences in the EEG? Well, basically, we did see differences between subjects with the low and the high memory quotient. Subjects with a high memory quotient, they revealed in this baseline condition as compared, so in the sham condition as compared to the baseline condition, they revealed over the left prefrontal region increased spindle activity, increased slow spindle activity, and also revealed increase in fast spindle power in the acute time period. So basically there was this modification, this increase in spindle activity that was more pronounced in the high MQ subjects than low MQ subjects. There was also a change here, however, in the opposite direction. So there does seem to be some difference between these groups, which, however, were differentiated, we have to say, then also post hoc, so after the experiment. So as an interim summary, we can say here that the subject group with high memory quotient that improved with SOTCS on the declarative memory task. And if we just look at the changes that these subjects had with high memory quotient as compared to low memory quotient regarding the task-induced effect, then they had increased spindle power in both the acute and the post-stimulation time period, which suggests that there is some kind of role of an inherent network state, which is affecting the susceptibility to slow oscillation TTCS. In order to look at things a bit further, and also because of other studies that revealed interesting results regarding the effect of oscillatory activity and how that related to memory consolidation, we then looked at the contribution of auditory stimulation, how that interacts with different brain rhythms, has the additional effect that one can specifically investigate the temporal aspects, because a tone comes at distinct time intervals and not as the slow oscillatory stimulation over, let's say, a 5-minute continuous period of electrical stimulation. So this is a study that we that brought our attention primarily to this topic, which you probably know. And here, just to get familiar with the graphs, here we have the grand average after tones during slow wave sleep had been given. That you see here, there's an increased evoked potential in the grand average. This was associated then with also increased fast spindle power, time locked to the slow oscillation, increased slow oscillation power, and there was an increased number of offline detected slow oscillations in the slow wave sleep period. And this was coincided then also with an increase in the retention of declarative memory. And what we had then looked upon in the study afterward was to see if we could more or less induce a kind of phase locking or quasi-phase dependent open loop stimulation. And so instead of applying phase locked to the first, I'll call this now the first oscillation, a tone, and then also to the second one, we first applied a pre-tone, let's call it, and this pre-tone induced an evoked potential. And the second stimulus we then applied when this evoked potential more or less reached its peak. And so this way we produced more or less a quasi-phase dependent open loop stimulation. And here again, there was this event-related response. Naturally, that occurred. There was not too pronounced increase in fast spindle power, actually not during the sufficient or during the adequate time periods. And however, what we did see was that there was an increase in the slow oscillation and spindle power in this 210-minute period of sleep per se. That was both significant for the slow oscillations as well as for the spindles. In fact, the spindles did not show an increase, they showed a decrease in power. And since there was this decrease in spindle power, we had thought, okay, then probably retention, or it could be that retention is poorer due to this quite strong decrease in spindle power. However, if we looked at the times where there was during the that the fast spindle power during the positive half waves, to what extent that was decreased. So again, sorry, if we look exactly at the how much spindle power was decreased during the positive half waves of the slow oscillation where they normally occur, where this phase locking usually occurs, then there was no difference between the stimulation and the sham conditions. And that one could say coincided with the fact there was no decrease in retention performance that we saw during our task. Furthermore, what the study showed us is that there was also a difference between when the spindle power was decreased. So we have within this time period here, within the sequence of stimulations, we have periods that are within sequence intervals. So basically when the stimulations, the tone bursts occurred, and then we have activity during this break period. And it was during stimulation that there was a significant difference between the fast spindle power during the within interval and the break period. And this is possibly contributed to a refractory period that the spindles, you cannot continuously increase spindle power, but there is a certain refractory period occurring after enhanced spindle power. And that is why there was such a strong decrement of spindle power in this intersequence interval. So basically showing us what information one can obtain of the network activity through applying stimulation procedures. In general, we find always two directions with stimulation procedures. One is to find out information about the network, and the other is more translational. So I keep checking the time. That's why I'm looking at my telephone. I will skip over this now and come to this question based on our study that I reported. Does slow oscillation stimulation efficacy depend upon EEG coupling parameters and slow oscillation slope in the control condition? Here, and this is an ongoing analysis, we employed this data in order to try to answer this question. And these are not the final results, but definitely they are actually final results. Yeah, they are actually final results, sorry. But presenting in a different context. So basically, what we did find is, and here to show what we're showing you, we have the phase coupling strength on the one hand, and then we also have the phase angle to which spindles are coupled to slow oscillations. And what it appears is that there are correlations that exist between either and that's not necessarily that new, that other studies have found things along these lines. But what we saw here is that the efficacy of the stimulation, that is to one extent one condition correlated with the strength of the phase coupling, and for another task, the efficacy correlates more with the phase. Essentially, what is so interesting here is that we have two different declarative memory tasks, the word pair associated task and the figural paired associate task, and that these two tasks appear to be more strongly associated with different aspects of the effect of stimulation. So that that indicates, which is actually quite normal, and where research is more ongoing, that there is a distinction in the underlying networks for different kinds of declarative tasks. Again, nothing excruciating, maybe novel, but on the other hand, it is in such a case that one does look more into the different network functions and how they relate to memory consolidation. We also found an association to the down to up slope of the slow oscillation that was apparent for the figural paired associate task. So this down to up slope of the slow oscillation, that is then where the period where the fast spindle activity occurs. So more toward the up phase, but from the more or less this down to up direction, down state to the up state direction of the slow oscillation. So, and then at the end, we looked also into structural aspects that could affect the efficacy of stimulation. Here, basically, we have not as yet looked into the efficacy of stimulation per se. We have, however, looked into how pathways, here particularly the pathway from the ventral hippocampus to the medial prefrontal cortex, to what extent that was then impaired. The memory consolidation was impaired if we impaired the function of this pathway, specifically during slow wave sleep. I believe I do not have quite enough time to go into this whole study. So that basically, I will just say that the impairment, the suppression of activity, specifically during slow wave sleep of this pathway, that did lead to a decrement in recent memory performance. So basically in this Barnes maze task, animals, they had to, because of their attempt to avoid the light, they learned based on the extra maze cues where this black or where this tunnel was. They were put on this Barnes maze, then they were put to sleep. During the sleep period, they were either stimulated optogenetically, there was an optogenetic inhibition of the pathway, or there was no optogenic inhibition that occurred over 4 days. And we measured in succession their performance on these 4 days. And then again, their performance on the 5th and remote memory on the 16th day. And if we just look at the performance on the 5th day, we see here that the stimulation animals did not anymore reveal this preference. There's a slight increase in the activity, number of head dips, the target hole, but this was not significant. Only on in the sham condition was there a significant preference on day 5 for this dark tunnel, for the location where this dark tunnel used to be. Now the maze had been rotated so that the animals could only go in the direction, go to the hole where they remembered, quote unquote, the dark tunnel to be. And coincident with this, there were also changes in the coupling between activity. mostly between the slow oscillation to spindle coupling and the spindle to shockwave ripple coupling. And as a final point of the talk, I would just like to mention, this is where we're also collaborating with Maxim, is that we are transferring this auditory stimulation paradigm to the mouse, where we stimulate during different phases of the slow oscillation. And here, results are ongoing. Here we are using an object place recognition task for this as a behavioral task. It does appear that performance is influenced by this auditory stimulation in the mouse. The electrophysiological effects there, we still have to continue with our analysis. And so with a summary, I would say that our data show that there's increasing need to consider the effects of baseline parameters, and that there are different differential mechanisms of consolidation for different subtasks of declarative memory. And there can therefore also be, or there is a differential impact of transcortical stimulation on different possibly population groups to be investigated, that there's a coupling between different rhythms that may react differentially, also dependent on the phase of stimulation. And lastly, that the effects of cortical stimulation procedures in the mouse, they may not be quite analogous to effects in humans, but that we will still have to see. And then acknowledgements. And yes, thank you for your attention. Whoops. That was not intended.
Okay. Thank you, Lisa. Very